and it's Celia Smith, yeah. Primarily my research is focused on deep water macroalgae and Halimeda meadows here in the main Hawaiian Islands. But a recent collaboration has occurred between NOAA, CRED, and HCRI, which has given me the opportunity to work with Peter Broom looking at algae and coral abundance around Lanai. So when I was able to take advantage of this opportunity, my first response was, well, okay, I kind of heard about NOAA CRED, but I really didn't know much about what they were doing. Who here, if I could see a show of hands, have actually heard of NOAA CRED? Okay, because so about half the people here have heard about them and know what they do. Well, to look at it in more detail, NOAA CRED is part of the Pacific Islands Fisheries Science Center, which is part of the NOAA National Marine Fisheries Service. And their basic objective is they have a number of different groups, and the Reef Assessment Group, which I'm working with, of the Coral Reef Ecosystem Division is involved in researching the biological organisms that are key components of the coral reef ecosystem. So very similar general objective to some of what HCRI does here in Hawaii. And the NOAA CRED Ecological Assessment main objective, so taking that goal and looking at it more specifically, is to document baseline conditions of the health of coral reef living resources such as fish and coral and algae and invertebrates in the U.S. Pacific Islands. That's a really big objective. To refine species inventories lists of these resources for the island areas, so try and figure out what exactly we've got going on there in terms of the inventory. Monitor these reef resources over time to quantify possible natural or anthropogenic impacts. To document natural, spatial, and temporal variability in the reef resource community. So if you go to one site, the same site year after year after year, are you going to see the same type of community there, the similar percent cover, the same number of fish, or does it change a lot? And they want to improve our understanding of the ecosystem linkages between and among species, trophic levels, and surrounding environmental conditions. So again, as you can see, HCRI and NOAA CRED have a lot of similar objectives and interests. Now, what's the deal with NOAA, CRED, HCRI, and me? Well, where I come into this picture is that in being in Celia Smith's lab, I'm a trained, or at least as well as I can be at this point, trained phycologist. And there's actually a number of phycologists in this room right now. And the caveat of being a phycologist, besides the thrills and joys that we have on a daily basis, is that when you go in the field and you want to assess the percent cover of algae, you can often take a picture and collect samples from that picture of what you're looking at. What that means is that from one small quarter-meter square area, you can collect 10, 15, 20 different species if you're looking at turf. So if you have all those species, you put them in a bag, you freeze them, and then when you get a chance, you go back to the lab, you unfreeze them, you pick through them in a microscope, you look at and try to verify what it is, you hope for reproductive structures, etc., etc. So whereas like you're an ichthyologist, you go and you count fish, you record the species, when you get up on the boat, you're done. But for a phycologist, the job has just begun. And as you can imagine, Peter Broom, who's with the NOAA CRED, is a phycologist and is in charge of meeting all those objectives that I talked about before. You're in charge of the Pacific Islands. What this means is that over time, you invariably have a backlog if you're one phycologist. So you can see some of this backlog here. So this one large Ziploc bag contains 12 smaller bags, which represent each individual photoquadra that was done with the surveying methods that they currently use. And so if you go out to sea for a few weeks, that few weeks could mean a year of inevitable field work or lab work to try and figure out what exactly they're looking at. So given the similar objectives between HCRI and NOAA CRED, I was being able to be hired to help Peter out with some of this inventory. So for the month of July, I've been working with Peter for the site survey around Lanai. And what this has involved is trying to identify all the algae that are in these little bags, so thawing out the samples and looking, and I'll tell you about some of that process, and also analyzing the percent cover of the photoquadrats taken. So my objectives in looking at Lanai, the reason we picked Lanai is because it turns out there was a certain amount of time that I had to work with Peter on this project, and when we looked at all the samples that are present in the main Hawaiian Islands, Lanai had 2 decent-sized bags, but that were small enough that we thought it would be a fairly quick project to complete given the time frame that we have. Also, I have some interest in Lanai in that for my own PhD research in deep water, we have a number of sites around Lanai that I've been interested in that we have some interesting results for. So I was inherently interested to see, like, well, gosh, what's happening in shallow water in these sites, and is it similar between the shallow water and the deep water sites? So I was interested to see what the results were going to be. So our objectives with Lanai are to first establish a species list of algae from 15 to 20 meter depths that were collected during 2005 and 2006. To assess the temporal and spatial variability in coral algal abundance represented as percent cover at these various sites around Lanai, and to interpret these patterns of abundance from an ecosystem perspective. So working with the other collaborators at HCRI, so the ichthyologists, the oceanographers, the fish folks, to be able to assess, okay, if we have one particular area of Lanai that in 2005 had a lot of fish, then in 2006 there was hardly any. Did it happen to be that there are trends with the algae that are present that, the years that you had a lot of fish, there wasn't much algae, or vice versa. So trying to use these different parameters to interpret these patterns of abundance with the algae. And are there possible differences in algal diversity among the sites? The sites that I'm talking about are represented by the 8 sites shown here. Sites 1, 2, 3, 4, 5, and 6 were surveyed in 2004, and in 2005, sites 3, 4, 5, 6, 7, and 8 were surveyed. So there's not exact replication from year to year. But what it means is that for sites 1 through 8, we can assess spatial differences, and for sites 3 through 6, we can at least assess year-to-year differences. There are also temperature sensors placed at sites 5 and at site 7 in both years that we will be able to use that data to look at any possible differences in abundance